Hey Bio, Mrs. Cozen, and we're still in Unit 3 DNA, and this is Video 2 Structure of DNA, and you are also going to have a picture that you're going to color and label. So yay for coloring, woohoo! Starting quick review of structure of a chromosome. We started to unwind some of your chromosomes in the lab that we did last week. We didn't get all the way down to this DNA, the double helix, the twisted ladder that you guys usually probably think of when you think of DNA. It's always at the end of the X-Men movies, if you guys have ever seen it in the credits. And so the DNA right here, this is your double helix. DNA is made up of little parts called genes, and then we're going to talk about all this little ladder stuff inside these genes today. And then we wrap those around each other, and then we wrap those around proteins called histones, and then we keep wrapping those just like a ball of yarn, and then it gets tighter and tighter until we make a chromosome. Last week we used soap and heat to unwind your chromosomes and to break up that lipid bilayer and then to break up some of these proteins. All right, so what does DNA stand for? DNA stands for deoxyribonucleic acid. Don't worry, I'm never going to ask you to spell that, okay? But that's where your DNA comes from, okay? Deoxyribonucleic acid. And we're going to talk about some of these different parts, this deoxyribonucleic stuff today. All right, so you guys make sure that you write this down, the definition of what we call a nucleotide. A nucleotide is the small molecule that makes up DNA. So right now, sorry, I'm going to rewind a little bit. We're talking about just the little part right here. See this little green thing I'm pointing to? We're talking about that stuff, okay? That's the molecule that makes up part of the twisted ladder, that makes up a gene. You put genes together, you get a double helix. You take the double helix, you wrap it around histone proteins, and then you end up with a chromosome, okay? So this part is much smaller than a chromosome. Sorry, let me fast forward to where we were here. So right here, you guys, this is the DNA kind of all flattened out. So think of that twisted ladder, but we've completely flattened it. And then you can see kind of a little more about what the structure looks like. You don't need to draw this part. You're going to get a model of that. You should already have your coloring sheet, and you'll be coloring in as we go along. This is just same concept, different model. You do need to draw this, however, okay? Make sure that you guys draw this right here. This is a nucleotide. This is the little structure. You guys can see it's kind of zoomed in. And when we put nucleotides together, we get our DNA. Okay, so that's the molecule that makes up DNA. And we call it a molecule. Remember what a molecule is. It's multiple atoms put together, okay? So we have a phosphate group. We have a sugar molecule. This makes up the outside or the backbone of our DNA. And the nitrogen base makes up the rungs of the ladder. Think the part that you actually grab onto to climb up the ladder. That's the nitrogen base. And we have 4 of those, T, A, G, and C. Okay, so make sure that you guys draw this picture of what a nucleotide is. Nucleotide is smaller than the DNA double helix. All right, so 3 parts of that nucleotide. If you guys drew the one on the previous page, you don't need to redraw this one. You can draw this one as well if you want to. It's the exact same concept, just different model. Okay, there's 3 parts. You've got deoxyribose, a phosphate group, and those 2 things put together make up the outside of the ladder, the backbone of our double helix. And then inside we have what's called a nitrogen base. Sometimes you guys will see it in our reading called a nitrogenous base. Same thing, base made out of a nitrogen compound. Okay, so 3 parts of a nucleotide. Either draw this picture, draw the one on the previous page, or you guys are more than welcome to draw both of them. Okay, now you guys have your coloring picture, which hopefully you guys already have glued in. Here's the exact same picture. I wanted to show you guys how the nucleotide here looks different model, exact same concept. Okay, so it says to circle and label 1 nucleotide on your coloring sheet. So for this one, we've got the deoxyribose, the phosphate, those make up the backbone, okay? See how these little stripes, each of the larger stripes represents the deoxyribose sugar, the smaller one represents the phosphate, and that means these little rungs on the inside, these are the nitrogen bases. So make sure you guys label these. You can label them D, P, and N if you want, or you can label them with the full term. Let me go back to our picture here. Phosphate, deoxyribose sugar base, and then over here, you've got deoxyribose, phosphate, nitrogen base, okay? Same thing here, deoxyribose, phosphate, nitrogen base. Those 3 things make 1 nucleotide. And then you guys can see we've got another nucleotide below it and another nucleotide. Okay, the nucleotides are our bricks that make up our DNA. All right, so we talked about the outside, the deoxyribose and the phosphate. Now a little more detail about the inside. Now, there are 4 nitrogen bases that we know of: T's, A's, G's, and C's. Okay, they're called adenine, thymine, guanine, and cytosine. From here on out, you guys can call them A, T, G, and C. Don't worry, I'm never going to ask you to spell these correctly. And these 4 nitrogen bases we represented when we did our little CATS demo at the very beginning of the unit. Remember how I gave you 4 letters and you rearranged them to make a lot of different combinations? Same thing with our 4 nitrogen bases. Creating different combinations of these in our millions of nucleotides that you would have on DNA makes the smallest bacteria is made out of the same 4 letters that you are made out of. Okay, the tiniest little algae is made out of A's, T's, G's, and C's. So is the largest blue whale. Okay, so these 4 things combined give us all the genetic diversity on the entire planet that we know of. Now, A, T, G, and C, if we were to find life on another planet, we don't know if their DNA would be like this. We don't know if they would have a nitrogen base. We just know we have a lot of nitrogen here. That's what we've evolved to use. And those A's, T's, G's, and C's only like to be with other certain nitrogen bases, and that's called base pairing. We've got a thing in the middle called a hydrogen bond. Okay, and it's only going to work between certain base pairs. A and T like to go together, G and C go together. Okay, the hydrogen bond holding the latter together won't work between like G and T. It won't work between A and C. It'll only work between A and T and G and C. Okay, so make sure you guys know it's held together with this little thing called a hydrogen bond, and that our A's, T's, G's, and C's connect directly to a thing called deoxyribose. Let's go back to our model really quick, okay? So here's our T's, A's, G's, and C's, and you can see they're connected directly to this thing that's labeled D. They connect directly to the deoxyribose, and then the phosphate connects the nucleotide to the next one. Nitrogen base, deoxyribose, nitrogen base, deoxyribose, phosphates connect them together. All right, so now let's work on coloring in this sheet. Okay, actually, I'm going to go back to this. So if we think T's, A's, G's, and C's, first thing I want you guys to do, I want you to use a pencil and I want you to label all the bases in the middle as T, A, G, or C. Everything that's got this little arrow pointed, let's call that a T. Okay, so write a little T inside this one, and inside this one, and this one, and this one, and this one. Those are all T's. Now remember the rules of base pairing. What does T go with? T always goes with A. So that means anything across from one of your arrows, label as an A. That's an adenine. Okay, so this one with the little Pac-Man mouth, okay, you can see that it formed, this is the hydrogen bond in the middle. So you can see there's 2 little hydrogen bonds. That only forms between T and A. Okay, and then down here you've got a T and an A, a T and an A, and a T and an A. Okay, it should be 3 hydrogen bonds. I'm sorry, it should be 3 hydrogen bonds. Okay, now, so we've got T's and A's labeled now. Now let's label G's and C's. Remember the rules of base pairing, G only likes C, okay? Our G ones, okay, is going to be this one with sort of like, it looks like a little open fish mouth here, okay? Let's call this guanine. Now guanine only pairs with cytosine. So if this one's a G, what's the other one? It's a C, okay? So this one's a G, this one's a C, okay? G, C, this one's a G, this one's a C, sorry guys, G, C, and G and C, okay? So that gives us all of our base pairs. Now when you guys are coloring, I want you to color all the T's red, I want you to color the A's green, the G's are purple, and the C's are yellow, okay? So repeat that, T is red, sorry, A is green, G needs to be purple, and C needs to be yellow. Then I want you to color your deoxyribose some kind of shade of blue, and then your phosphate like a light blue or a teal, okay? If you can't find light blue or teal, just use something different. You could use like pink or something for phosphate, okay? So we've got a nucleotide circled and labeled. We have base pairing talked about, and then you guys have all your nitrogen bases colored in. So this is going to be the end of video 2, and we're going to have to call it video 2A. I'm going to have to record a video 2B because I am out of time.